Welcome to 4IR. Biomanufacturing technology utilizes living cells or microorganisms to produce commercial products like vaccines, biopharmaceuticals, and biofuels, relying on techniques like genetic engineering and fermentation. The Biomanufacturing Industry Development Center at the CSIR is a first of its kind in South Africa, and it aims to support small, medium and micro enterprises involved in biomanufacturing in meeting their customer needs within short time frames and be able to exploit market opportunities.
My name is Tsepo Tsekwa. I'm a research group leader and chief researcher in biomanufacturing technologies at the CSIR's chemicals cluster. My experience growing up, I think in this context I was quite fortunate in that I found myself doing quite well educationally. I did very well in maths and the sciences, and I think it's the experience of most of the team here. We found that, you know, in the context of the primary school I was at, I was quite at the top of the class, and so I think that that instills a lot of responsibility and pressure as a young person, because then all eyes are on you and their hopes and aspirations are looking at you to succeed. So that experience, I think, stands out. I was a little bit fortunate. I also experienced school in the UK at high school level, where I think it was quite a diverse North London-based school, which was quite an eye-opener for me as a young person. But I managed to keep up and managed to do quite well, similar to what I had experienced at primary. The thing with a young person's mind, you find yourself doing very well, and the sciences and mathematics and STEMI subjects, as we call them now, are typically subjects that are considered to be quite difficult. And the children who tend to do well in those subjects are normally encouraged to keep working on those subjects throughout their studies. Then you find yourself making choices in subjects as you head towards completion of high school that focus you towards a more scientific career trajectory. In my context, I happen to have a keen interest in human health. In the end, I didn't become a doctor, but I ended up in a field of life sciences that does interface with the healthcare space. So I had pursued these subjects with a view of going into that broad healthcare related space. How has the field evolved over time? So I think that biotechnology in general is a very fast moving field of study. I think that one of the key things that have resulted in rapid evolution is the discovery and development of recombinant techniques, or techniques that allow us to manipulate DNA, to manipulate genetic information in living systems in a simple and accessible way. The development of techniques that allow us to take genetic information from one organism to another, the ability to take those genes and produce proteins as products in different systems, the ability to study that information through different ways of sequencing it, have allowed us to now extract vast amounts of information. So despite my training having been, I finished my PhD in 2006. That's a few years back, but what we've seen as a trajectory in the field is the entrenchment of these recombinant DNA techniques and their evolution now to what people are calling synthetic biology, and the ability to now in a much more facile or simple way, manipulate large sets of sequences and move them around and exploit them, so to speak. I think that's the main evolution I see. You might be aware that in the past few years, this very same technology I'm talking about has seen recognition through the Nobel Prize, for example. You might be aware actually that even today, some of the medical technologies, for example, monoclonal antibodies, which is an area of work that I'm quite passionate about, has been facilitated by the ability to take genetic information and coding those molecules and using it to then produce them in a larger scale through these molecular techniques. So I think that evolution is quite prominent and key in my mind. The other aspect I can mention is that, in our context, in South Africa, we do science, but at the CSIR, we want to do science to improve lives, right? And the key evolution in my field, in the context of our work at the CSIR, is that we want to use these fascinating and exciting technologies that I'm talking about, but we want to apply them now to producing value. To producing value in the sense that we are able to now improve lives through impacting socioeconomic development. In our case, I can share examples of our work having come from the laboratories we're in now and having been transferred to form a new company that at its peak was employing, quite a number of people. That's impact, and that's really quite heartwarming to see. In our other context, we are looking now very closely at the idea that in South Africa, within our shores, we'll be able to now manufacture and produce things like biopharmaceuticals and vaccines, and that's a key part of the work that we do, so that we start to see and start to develop a national sovereignty, a national capability that allows us to make these things in the country. I think that's an important evolution to say that as the technology improves, we also want to strategically position ourselves to use that technology to improve the lives of South Africans and to make impact, and hopefully to also develop the economy.
Hi, I'm Sibongile Mtimga, a postdoctoral researcher here at the CSIR Biomanufacturing Technologies Group. I think it's actually quite a funny story because growing up, I convinced I wanted to be in the consumer food space. So my principal was like, no, not with your marks. If you want to learn how to cook, we'll make a plan. And then that's when I ended up signing up for sciences in the grade 10 and had extra classes to do the cooking that I wanted. And then I think whilst doing that, I realized there's actually a lot of science that actually comes into baking and cooking as well. And then I ended up actually enjoying both worlds, but I liked life sciences more. And then I did that, and then when I moved to varsity, I did human anatomy physiology, which was medical biosciences, and it included a little bit of medical micro. And I liked that because it had the health aspect of it. I liked seeing or understanding what causes some illnesses and diseases and what could be done to actually treat those. And then I think from there, that's when I kind of like, okay, I think I want to be in this space, but I do not want to be a medical doctor. So after I completed my undergrad, I was fortunate enough to be offered an internship at the CSIR, which I started, I think, around about 2014. I joined a group that was more focused on biochemistry, which was like, okay, it's an elective, but let's see. And then I must say I learned a lot. I actually had good supervisors, good mentors that actually showed me the life sciences that I enjoyed is actually involved in biochemistry. It's just now we're using both chemistry and life sciences. And with that, I could actually do what I've always found interesting, to actually come up with solutions on how to treat certain things, or how to move around, how to disinfect better, and people would actually get healthier. And then through that, I think I moved around. I focused mainly first on using metagenomics. Metagenomics was just a process where you extract whole genomic DNA, but in the end product will get you to a certain, or your target protein. And then over the years, I've then grown because I've been around. Right now, I'm actually awaiting my graduation, my PhD graduation in biochemistry. The message I have for the young generation is that solutions to most of our problems are actually within reach. They are within us. We just need to align our capabilities and our interests in the right spaces. Then working together as well, we can actually achieve a lot, and we can get a lot done. With science, engineering, everything, if we align our skills, our skill sets, we could come together, solve a lot of problems. What I can't do, the next person can probably come up with a better plan on how we could address certain things. So we just need to align our skills, work together, and we could solve a lot of problems.
Zbongile Mtimka will now show us a demonstration of how monoclonal antibodies are produced and how target proteins are isolated.
So namhlanje silapha CSIR, we're in the biomanufacturing technologies group, where we'll be showing you our daily routines on what we do. So as my colleague earlier, Dr. Tsepo, had alluded into the things that we look into making our monoclonal antibodies, there are different systems we use. One of them is this right here, which is a plant. It's from the tobacco family, Nicotinia benthanium. So you can smoke it. We just use it to manufacture our target proteins that we want. So I'll show you from when we grow them, up until it's at a stage where we can now then use it when the target proteins and monoclonal antibodies. Yes. So we have what we call a conviron. It's our growth chamber where we grow them. So this here mimics the different conditions where you'd grow the plants so that they'd grow to your optimal temperature. So manifestos is one and now the same thing. Then we will grow them. After a while, we remove it as cool it to a point where we can now then use them. And then when we use them, what we do is, like humans, we have an immune system that if you introduce anything foreign, your system wants to get rid of it, then you can heal yourself. So what we do in the plants, so this is when we manipulate the DNA and then we transform it into cells, then we then have to make it in a liquid form that we can then inject here. But then what we do is we hijack the plant's mechanism to make it produce more of the target protein, your monoclonal antibody that you're looking for. And then it will then, after a couple of days, when you're done actually infiltrator, you put it back in a different chamber that has bigger plants. Then after a couple of days, CSI coupe, then we will go into the process of processing it, which I will also show you later. But what I'll show you now is how we then infiltrate the plants. If you have I syringe some. There you are, my needle. So to introduce this material into the plant, you have to inject, like poke holes in it. So what we do, I prefer infiltrating from the back, but bakhona banza ngaphambili. So what you do is you just make small holes, then it opens up the plant, then you suck up your mixture, not a great smell. And then what you would do is then press it against, and then, then as you infiltrate, you will see that your actual leaf looks wet. Then it means it's getting into the plant. Then you are introducing the infection, that's how you then call it. And then when you've infiltrated the whole plant, then we move it into the conveyor where we would keep them. And like I said, these chambers will mimic the growth, like the setting where you'd have your plant, a certain light level and then dark level, and also the temperature and the humidity. So then you put it in here, but right now it's at the dark cycle, which will then change after 6 hours to light cycle. Then after a couple of days, when we know it has produced, the plant's mechanism has made what we want, we would then extract, because we need to get it out, right? So there's different ways of extracting it. We just remove the leaves, and then we have a blender that we use, but then this is how it would look. So you can imagine if you have a spinach, when you chop it up, you'll also get that green juice at the end. So your protein will be there with other proteins from the plant itself, but you can't then now use that to say treat something. You need to make sure that it's clean, clear, and just has your target protein. So from here, you would then have to separate the leaves material, your solids, from your liquids. Then when you have your liquids, then my colleague, Dr. Pillay, will then take you through the process of purification. So Dr. Tsepo alluded to it as chromatography. That's where then you would separate them based on their molecular weight or sizes. They would separate in using gel electrophoresis, and then you would then be able to get your specific target protein. Then from there, the formulations happen and characterization, which Dr. Cindy Buthelezi on the other lab will show you.
Welcome back to 4IR. Today we're at the CSIR to learn more about biomanufacturing and the world-class science that goes into this fascinating industry.
Hi, my name is Priyan. I'm a senior researcher here at the CSIR within the technology demonstration group led by Dr. Tsepo Tsekua. Growing up, I was always fascinated by biological systems. You know, I was a kid who always liked to play outside, and I would be very fascinated by just living things, and I think that resonance with biological systems and really biology and science led me to take up a vocation within that field. CSIR is a premier world-renowned scientific organization. And so, having joined the organization as a young postdoc has really put me on a trajectory to be in the place of most potential, and I'm surrounded by really exemplary scientists, like Dr. Tsukua and Dr. Mtimka and my colleagues within the lab. And being in that environment, has really enabled me to come into my own and be part of a team that is working on so many breakthroughs that we hope one day will make a significant impact within the lives of South Africans. So I think CSIR, I'm very grateful to the organization for providing the conducive environment for this growth and development within young scientists.
Senior researcher Priyan Pillay will demonstrate the next steps of monoclonal antibody production and what equipment is used.
And as my colleague Dr. Mtimka mentioned about the process of agroinfiltration, now that has taken place over a 7-day period and now we are ready to extract the protein from those leaf plants. And so this is how we perform that extraction and purification. We use what we call is an Acta Advance 150, and this is an instrument that automates the process of purification. Okay, so you can see here that there's various lines that go into our extraction buffers, and they connect up to the machine, and over here you'll see all of the different pumps that pump the solution from these bottles into the machine from our sample, and it purifies our sample in a sequential manner. Now here's a great example of a column, and so this is a column that's used for size exclusion chromatography. It's a column that separates molecules based on their size, and so this column is used for polishing molecules up. So after you do an initial capture using affinity, for example, you may want to purify the molecule even further, especially if it's for therapeutic use, and so you'll use a column like this to further purify the molecule based on size. And so what you'll see is basically once you've hooked up your sample to the machine, so here we have like a sample pot here, right, and you will hook up the sample line to this sample pot over here. So you'll see here, this is our sample. So for example, S2 will be for sample. So you can take this line here from this, and you can hook this up to your sample here. Basically, once that's hooked up over there to the machine, it's basically going to pump this sample into the machine, and it'll purify your protein from here. Okay, so this will go into the machine, and we will run a series of buffers through that, and we'll be able to purify whatever protein is inside this sample over here. And that's how we purify proteins on a large scale. And this process can be used to purify antigens. It can be used to purify vaccines and biopharmaceuticals. We also use it to purify enzymes as molecular reagents as well. And this is just a summary of that process. Pursue your passion. I believe that if you're passionate about something, whatever it is, it could be science, it could be engineering, you'll make a success out of it. I think that our youth of today really has to reflect on their contribution to society. We look upon them as the stars of the future to help us improve society. There are so many people within our country that really need their help, and so it is our responsibility as their mentors to help them and guide them into becoming the best of the best, and the bright future of tomorrow. And so my message to them is to pursue their passion, and they will make a success out of it.